Hi, my name is Erika DeBenedictis. I'm a postdoc in David Baker's lab, and today I want to sort of give an overview of how the Baker lab uses deep mutational scanning data as part of protein design. So as a general outline, I'm going to talk about the de novo design pipeline in general, and in particular, how we design binding proteins to novel targets that we may not have examples of binding proteins for in the PDB. That's a pipeline that I think is really nicely representative of how a lot of the protein engineering projects go in the Baker lab. And in particular, it's a really great example of how people use site saturation mutagenesis data in order to understand and improve the proteins that they've designed. And I'll talk also about some of our sort of ongoing and future efforts to use this sort of data. Okay, so to start off with, backing up a minute, I think a lot of people at this symposium have talked about using deep mutational scanning data to understand proteins or genomes and functions. Here, the Baker lab, in addition to wanting to understand how proteins work, also wants to use this data in order to improve our own design tools and improve Rosetta or other models that can assist us in building proteins that we want that there may not be examples for that occur naturally. And one sort of category of proteins we might want to produce are binding proteins. So when COVID-19 occurred a year or so ago, we knew that this was a virus that has the COVID spike protein that is able to enter cells by binding to ACE2, which is a surface protein. And one type of protein you might want to make is a protein that prevents that interaction from happening by binding to the COVID spike protein in the ACE2 binding site and preventing the spike protein from binding to and entering cells anymore. And the Baker lab was able to quite rapidly engineer some small proteins that successfully and very strongly bind COVID spike. The sort of traditional way of doing that is to find an example of an existing protein that binds the location you want. In this case, that was very easy because we know that ACE2 binds the spike protein. We know it binds in exactly the right spot. And that allows us to take that PDB and steal the binding interface from the ACE2 spike interaction and then graft it onto some other sort of scaffold protein that may have better properties like being smaller than ACE2 or more thermostable, and so on. Lots of previous work has sort of done this trick of doing what's called motif grafting, which is identifying a natural binding interface and using it to create a new protein. However, the COVID example is a little bit special in that perhaps the next time there's a virus or the next time we have some other biomedical application, we won't be so fortunate as to have an example of a crystallized binding protein that is in exactly the spot we want. And so we'd like to be able to move beyond needing the natural example of a binding interface and be able to make up our own binding interfaces. So in the last couple years, the Baker lab has been improving our ability to design binding proteins for which we have no natural examples of binders. And the way this works is you begin with a library of small proteins that you might use as scaffolds. And then you test to see whether those small proteins, any of them in any configuration, could possibly have good geometric fit with the position you would like to bind on your target. And once you've identified some docks that look reasonable, then you can use Rosetta to design the interface residues between the target protein and your mini binder in order to arrive at a small designed protein that we hope binds the target protein. And at the end, the way this pipeline works is people generate millions of these sort of putative design models that imagine a binding protein that might work. And then you filter it down to a few tens of thousands of designs that you sort of think have the best properties based on our just best understanding of what works. And those get synthesized on a chip. We're very fortunate that the proteins we're making are super small, so they're usually 70, 80 amino acids long, so you can chip synthesize them in bulk. And then we use yeast display and NGS sequencing in order to quantify which of all of those, straight out of the computer proteins actually has binding activity. And in fact, most projects do this sort of synthesize, screen, measure pipeline 3 times. So the first time, you do an initial screen of designs that are straight out of the computer in order to figure out which of those designs has any activity at all. The second time, you take those initial hits, you do a site saturation mutagenesis library in order to sort of characterize the protein and figure out if there are mutations that could improve it. And the third time, you make a combination library of the beneficial mutations in order to produce a sort of like final design that has the best properties that it can have. And this pipeline is pretty good. After people come up with these optimized combo library proteins, you can express them in E. coli and quantify their KD quite accurately with like an octet in vitro. You can also use CD spectra and other techniques in order to figure out whether the secondary structure the protein actually adopts matches what you imagine. And people have had very good success, both designing proteins that bind strongly and also seeing that they validate both in CD spectra and crystal structures. So for example, the COVID spike binders are like picomolar binders, which is quite good. There are many people in the Baker Lab who work on binder design projects. So there's many people to thank for the success of this pipeline, but in particular, I just want to highlight Longqing and Brian have in particular been working on improving the hit rate, designing binders for which we have no natural examples. So this is just a great graph that shows that even as recently as 2018, people would order 50,000 designs and none of them would have initial activity. And that has very much become not the case anymore. The hit rate for the initial screen step of the binder design pipeline has risen considerably to the point where you can pretty reliably get initial hits for targets that have reasonable properties. So this is a huge success story, and this is going to be written up soon, so you can all sort of hear more about this. In particular, I want to highlight how the Baker Lab uses site saturation mutagenesis data as part of this pipeline. And I'll talk about 3 ways that we use it. First off, to validate whether or not the design model, which came out of the computer, is correct remotely. Second, which I think is more similar to what pretty much everyone does with this data, which is to design multi-mutations of a particular protein that have better activity or properties. And finally, and this is like sort of another more unusual application, sort of the final thing we do with SSM data is we try to use it to identify problems with Rosetta itself in order to diagnose and fix them. So not only are we trying to learn how the proteins work, we're trying to make our own scientific computing software more accurate using this data. So here's an example of one of the first places that the Baker Lab uses the site saturation mutagenesis data. So in the upper right, I'm showing the design model. So this is a 4 helical bundle that has been docked to the gray target protein. Helices 3 and 4, we hope, are participating in the interface with the target protein, helping it bind. And the rest of the helical bundle, the interior, the core, is holding the protein together into this fold that we imagine and hope it adopts. And this is an example of SSM data that supports the design model. In this heat map, the blue squares are indicating that a particular library member is de-enriched, so you can't make those mutations without ruining the protein. Conversely, red squares indicate that a mutation is the same as or beneficial to the function of the protein. So you can see that there's sort of 4 regions in this heat map corresponding to the 4 helices. Helices 3 and 4 on the right, you can see there's sort of 2 bars of dark blue area, which correspond to those 2 helices. And the reason kind of the whole thing is dark blue is because you largely can't play with the residues in those 2 helices very much without breaking the protein, either because it stops binding on the outside of the helices, or because if you make weird mutations to the inside of the helices, the protein will stop folding right. Similarly, helices 1 and 2, you can see there's this sort of characteristic every 3 residue pattern where the interior residues of those helices are important and you shouldn't touch them, because if you mutate them, that helix may no longer fold properly. And you can see exactly where those blue residues are in this design model. They're every 3 on the interior of the helix is important. So this is data that everyone hopes they will get when they run an SSM on an initial design that has worked. This SSM data supports the idea that our small protein both folds as we imagine and also binds using the interface we designed. So this is great, and people would move forward with this one. Conversely, here's an example of SSM data you hope to not get. You can see here now we have a 3 helical bundle. It's binding the gray target protein, and the distribution of mutations you can make to this protein is basically random, or at least it doesn't correspond to the design model we imagine. So this data doesn't support the idea that this small protein folds this way or that it binds using the residues we designed. And what's interesting is that the Baker Lab does find binders like this one that definitely bind somehow, but not in the way we imagine. We are interested in figuring out how they actually bind, but in general, people usually will find this sort of result and then discard this particular hit from sort of further analysis. So in this way, the site saturation mutagenesis data gives us a first look at whether or not our design model is remotely reflective of reality. Now, I showed you this graph a couple minutes ago. This shows over time the hit rate of the initial design round. And we can see that we've gone from basically a 0 hit rate to a definitely non-zero hit rate, which is amazing. What this doesn't show is the fact that some of these initial hits are not going to validate. And so that's what this orange shows. So this shows what percent of the initial screen hits for which we order site saturation mutagenesis libraries actually end up being correct. And you can see that it's not 100%. So, half-ish, a little less than half-ish of the design models that we create that do bind are not binding for the right reasons. So I really like this graph because this gives us another metric that we can try to improve upon. Not only do we want to raise the absolute success rate of finding a binder, we want to raise how often we find a binder that is the binder we wanted, rather than something that's binding for some other reason we don't understand. The next thing we do with it is when you do get a validated design model, you can then design a combination library to check to see if a few mutations to this protein would cause it to work better. So this is an example, once again, of the validated model that I showed you 2 slides ago. Here I've highlighted a few of the residues that we might include in a combo library. So in particular, we would look at residues that seem to be quite important. So they're in the core or the interface of the protein, and that have just a few possible mutations that seem to improve function. And we would create a combination library that allows us to check whether combinations of those residues together cause further improvements. Now, the third way that the Baker Lab uses this site saturation mutagenesis data is in order to try to identify and fix systematic problems with Rosetta. And I think this is so cool. Shout out to Hugh, who's a postdoc who has done really amazing work in this area. This is all sort of his design. This is a very clever way to go about it. One of the first things that we think goes wrong when a binder doesn't bind is it's possible that the small protein we've designed just like doesn't fold stably, or it folds into some other conformation than we think it does. And there's been lots of work. Gabe Rocklin, sort of in the a few years ago, worked on this quite a bit as well, to try to figure out how to predict whether or not a protein is going to be stable. And the best thing people came up with was sort of this graph, where you can predict the stability of one of these small helical bundles, and you can measure its stability experimentally, and you see there's a correlation, right? And there are also outliers. And Hugh was interested in the idea that perhaps these outliers are not so far away from a functional protein, and that it would only take a few mutations to rescue these designs. So he went and he picked a few of the designs that are sort of in this box, things that we think should be stable, but don't end up being. And he ordered site saturation mutagenesis libraries for them. And here's one example of what he came up with. We see that in general, you can't mutate much about this protein and have it still be stable, but there's a few mutations that do rescue it. And in particular, one trend he noticed was that there's a few instances in which you mutate large to small amino acids. And this trend appeared actually in several of the different mini proteins that he tested this way. There were a few instances in which it seemed like large to small amino acid mutations helped, maybe in general or in many cases. And so he went to go try to figure out, like, why is it that Rosetta is designing proteins where all you need to do is change large to small amino acids? And so he looked by hand at some of the particular examples where large to small helped, and he found that it looks like there's atom clashing. The atoms are a little too close together. And he quantified this amongst many different designs, and found that if you look at the histogram of the distances between certain atom types, you'll find that Rosetta designs proteins sometimes that are really too close together, and they are closer together than they might normally be in natural proteins. So you can see the orange histogram here shows the designs that exhibited these large to small rescue mutations, whereas blue are natural proteins from the PDB that shouldn't exhibit this. And so we can see there's quite a few atoms that are a little too close for comfort here. And what's very cool about this is this was very easy to fix, right? Hugh went into Rosetta and changed like one hash define to alter how far apart atoms needed to be for Rosetta to weight it to have reasonable energy. So this was a very cool example of using this sort of data in order to identify and fix like an underlying problem in our computational pipeline. And finally, before I wrap up, I want to give kind of a brief, just a brief mention to the ongoing work we have to use deep mutational scanning data more holistically in the Baker Lab. And there's kind of two sides to this coin. One side is trying to collect better, really high quality data sets. And the other is to use machine learning and other models to operationalize that data in order to improve the hit rates of all steps of this pipeline. So I'll talk about the data sets first. So we have an ongoing collaboration with Will Greenleaf's lab at Stanford. They have developed a high throughput method for doing in vitro binding curve experiments. It's a very clever assay. Basically, you do an in vitro binding curve assay inside of a MySeq flow cell. And the trick is that you can actually just sequence that flow cell ahead of time in order to figure out the position of all million library members that you've put on that chip. And then when you do your microscopy experiment, you already know which library member is in which position in your image. So you get the ability to measure somewhere between 100,000 and 1 million different small binding proteins in a single 2-day experiment. And why we're interested in this data for a few reasons. First off, it would be great to have an in vitro data set to complement the yeast display data we already have. Yeast display has its own weird experimental quirks, and it would be great to have just like any other assay to measure it against. And then in addition, the way the Baker Lab has sort of traditionally run yeast display is very much designed to help us identify the top hits, but not really designed to quantitatively tell you how strongly all of your library members bind. And so gathering this new data set would give us an opportunity to accurately measure like medium and weak binders in a way that would be a really valuable resource. And finally, we're interested in using this data set as well as the yeast display data and many others in order to build computational models that help us raise the hit rate of the initial design phase, how many of those validate with SSMs, and also just in general, how good are the properties of these binders that we get at the end of this pipeline. And we've been really interested in following all the really cool stuff that's been happening with representation and transfer learning in the past couple years. And so far, we've had some success with using learned models to create essentially better filters that are a little bit more predictive of which computational models of the millions that are produced should actually get synthesized in DNA and tested experimentally. So we're expanding a lot in that area. We have a new subgroup called Machine Learning on Experimental Data. And if you're looking for grad school or a postdoc, you should come talk to David if you're interested in that area, because I think there's a lot of interest there and we're expanding there a lot. And I want to also acknowledge that the Baker Lab is a huge group. There's more than 100 people, and in this talk, I only scratched the surface of what goes on here and mostly highlighted work by my colleagues who have been improving these methods for a very long time. In particular, I highlighted a lot of Brian, Hugh, and Longqing's work on the binders and understanding Rosetta pathologies and fixing them. But there's many others who have made this work happen. So thanks to them. And thank you so much. And I guess it's question time a month from now when I'm recording this.